FIIRO. I'm a medical laboratory scientist and I came from a State University, but I work with Wakamedi's Healthcare Limited, which is a medical mobile diagnostic company. Okay, so Wakamedi's Healthcare Limited is actually a medical mobile diagnostic company, as I said, and we are looking at exploring molecular diagnostics because we do basic chemical pathology, hematology, microbiology analysis. So we are looking at starting up the molecular diagnostic aspect of the laboratory. So we actually thought it's good for me as one of the core scientists in the company to get trained and acquire the skills for us to be able to set up our laboratory and also be able to train other persons who are going to be employed afterwards to work in the molecular laboratory. The workshop has really been amazing. You know, when I came in initially, I actually had a thought that it was going to be maybe 50% theoretical and 50% practical, because I know that the reagent for molecular diagnostics is actually very expensive. And to my greatest surprise, I just realized that we had everything, like 70% was hands-on. So it's not hands-on that you're working with your group members, it's hands-on that you're working on your own. So everything is made available for each participant, and it's been amazing. Because at this point I know that I'm not just going that I have the 50% knowledge. I'm not just going like a startup. I'm going like someone that is skilled. I'm able to do it on my own, from the agarose gel electrophoresis, starting from even the DNA extraction to the agarose gel electrophoresis to the PCR, which we did. LDS page, I've only heard about it in my 4th year in the university, and to my greatest surprise, I did it myself. Western blot, which is what we're going to round off today. It's been amazing, and I know the workshop actually met with my expectation. And I actually didn't even know that I can actually bring my own samples here to analyze subsequently, maybe when I start my own master's and PhD in view. So I just think that FIIRO is doing a whole lot of work that people need to know out there, because if I didn't know, a lot of people didn't know, and I'm actually saying this to people outside there. FIIRO is actually open to people that are doing research. If you have your research work or your PhD to do, it's open. All you need to do is just collaborate with them. They're open to partnerships. So FIIRO is an amazing place, and I'll be glad to come back soon.
I came for the workshop for 2 reasons this time around. One, I'm a prospective PhD student in the University of Lagos, and my interest is in forensic genetics. That will require a lot of molecular biology skills. And the second reason is I'm thinking of starting a molecular biology lab with time. At least I have a timeline to that within 5 years. And at least coming around here is to look at what I will need to set up such a molecular biology lab. This is my 3rd time of attending this kind of training. The first one that was organized 2017, I couldn't attend for some reasons. The second one, of course, I was here at FIIRO 2018. The third one, when FIIRO collaborated with NIMA, I was there at NIMA all through the training. And then this 3rd time, at least, now that mind is strong to set up your own lab and then pursue your PhD. Actually, I'm happy that I'm here because consistently now, out of those 3 trainings, the first one I got DNA. The second one I did not get DNA. That is during the hands-on training section when I handled a lot of equipment myself. And this 3rd one, at least I got DNA. In fact, I actually have an intention to come again next year if FIIRO will organize another one.
I'm Dr. Mrs. Fola Shadi Olajuyi. I'm an associate professor of biochemistry from the Department of Biochemistry, Federal University of Technology, Akure. I've been a grantee of many funding bodies, and I just recently set up a molecular biology laboratory with the research grants I won. So I have different equipment. Do I have a postdoc monitor? Using the equipment and also pay the students. But I wanted to have hands-on, experience on the equipment in my lab, like the PCR machine, the gradient and recycler. I need to know how to use and also to troubleshoot. So this was one of the reasons I came, that even when my postdoc or PhD students present results to me, I can really understand what they have presented, and I can also use the equipment myself. and have really done well, especially in the participant-instructor ratio. It's been excellent. You know, I can say this is really an hands-on workshop where every participant really participated. You know, at time you attend a workshop, it's not all the participants that are participating. They are there, they are attending. But in this particular workshop, all the participants, I can say, participated because we had everybody laid hand on every experiment, and we asked questions. Also, the questioning, I really like the idea of the quizzes, you know, that were given during the lab session and even the theoretical section, the lecture session. We had quizzes, we had homework, we had tests, you know, all these things broaden your skills and also help to retain the technical details that we have really learned from the workshop. So it's been a fulfilling one. I've attended both locally and international workshops, and I can say that this one can compete favorably. So it meets global standards.
My name is Aruna Isiaku Umar from Federal University of Technology, Akure, Department of Biochemistry. I'm a laboratory technologist. Since 2011, I've been exposed to molecular biology training. So anywhere there is an opportunity to attend this type of training, I always want to attend so that to gain more knowledge to build on what I have learned over the years. The impact is immeasurable, I would say. I've attended workshops on molecular biology. I could spot the difference. This is unprecedented. I'm very happy the way the organizers handled the training. I was thinking it is just the normal lecture, and a facilitator in the practical, in the laboratory will just come in front of the participants and be demonstrating how to carry out DNA extraction, PCR, Western blotting. But in this case, every participant was given a sample to analyze themselves, extracting DNA, extracting protein, running a PCR for the gene, and doing Western blotting. It is unprecedented. Me coming to FIIRO to attend this wonderful workshop gives me a rebrand, a renewed perception, the real perception of FIIRO, that FIIRO is actually working. And I give kudos to the teams in FIIRO. They are doing well.